Alright guys, so first time trying this style of teaching, so we'll see how it goes. Today we're going to be looking at genetic data, having a look at number one, what sorts of things can we look at, and then number two, how do we record and measure them. So you can see the heading up there, syllabus point 543. If you haven't already, pause now, get your notebook out because you are going to be writing a couple of notes along the way as we go through this. Here's the syllabus dot point. If you ever look at it, it's a bit of a strange syllabus dot point because if you look at what it says, collect, record and present data to represent the frequencies of characteristics, it's telling you you have to do something, and it's not really saying anything about the theory you need to know. So really this is a practical dot point. In the second part of this lesson, you guys are going to be doing a bit of research to meet that requirement. But if you look at what we need to collect data on and understand data for, we need to know what frequency data is, and more importantly, we need to know a little bit about this thing called a single nucleotide polymorphism, or a SNP. So we're going to be learning a little bit about both of those things before I send you off to collect that data. So the first slide here is key terms. Remember, you can pause this video if you don't get a chance to write all this down. So just running through a couple of terms that we definitely need to be familiar with for this section of the syllabus. The first one is population, which we talked a little bit about in year 11 when we did ecosystems. Essentially, populations are just groups of organisms that are capable of breeding together and producing fertile offspring. So if we look at an example of something that can't be a population, it might help to make it a bit clearer what a population is. So for example, horses and donkeys are capable of interbreeding, and they're capable of having offspring, and you call the offspring of a horse and a donkey a mule. But all mules are born infertile because horses and donkeys are different species. And so therefore horses and donkeys can never make a population because they are not capable of producing fertile offspring. On the other hand, if we look at a more familiar example with human beings, we have all of these different cultures and races around the world, but all human beings are capable of interbreeding with each other and producing fertile offspring. And so as a result, we can look at any group of humans as a population. And then if we use population, we can go to that next dot point, population genetics, which is just over here. And population genetics is the study of how populations change over time. So we're just looking at genetics, and we're just measuring whether things are becoming more and less frequent over time. The gene pool is the total collection of all the genes in a population at any point in time. And we use the gene pool to measure things like biodiversity by looking at how closely related organisms are, or how distantly related they are, how much variation there is between organisms in a population, whether organisms are all homozygous or whether there are recessive traits in populations, those sorts of things. And the last key word on here is frequency of alleles, and frequency, as the name would suggest, and like you've learned in maths, is just looking at the total number in proportion to the population. So a classic example for humans that you could talk about with frequency is red hair. If you look at a country like Australia, carrying that red-haired allele, or showing it at least, is quite uncommon. There's not a lot of red-headed people in the population in Australia. And so therefore it would have a low frequency. But if we went over to somewhere like Ireland or Scotland, we'd find a lot more people with red hair in those populations, and therefore the allele would have a greater frequency in those populations. So the first thing I've got up here is a little graph of blood types. So you can see over here on the left at the top we've got type O, and then type A on the right, and then down the bottom type B. And if you just have a look at that as a snapshot, you can see first of all there's very obvious and very different patterns. The O and the A blood type sort of have more similar patterns than the B blood type, and that's because B blood type is the rarest blood type, and so it is the least frequent around the world. If we start up at the top left with the O blood type, you can see that the creamy white color means there's a very high proportion of people in the country with that blood type. Oh, sorry, very low proportion of people in the country with that blood type. And the blue means lots of people have it. And so we can see that South America and Mexico and the bottom of the United States, very high proportion, nearly 100% of people. Also another group up around Alaska and Canada. Whereas over in countries like Eurasia and Russia and Upper Eastern Europe, it's less common. And kind of an inverse pattern is visible in this B blood type population map, where we can see that B blood type is quite rare in countries where O blood type was common. So remember that the O blood type is actually a recessive blood type, but it is also the most common blood type in the world. And the A and B alleles are more recent mutations in human history that are starting to become more populous in the world, but they spread quite slowly. And the key point to look at here is that this is an example of us being able to look at a genetic trait, blood type, and we can use that to look at trends in populations. So I can look at this graph or this chart, and I can quite clearly say, well, B blood type is very uncommon in Australia. O blood type is fairly common and very common in the top end of Australia, and that's because indigenous populations all had type O blood. And then the A allele is moderately common, and again there is a clear pattern. Areas that are coastal on the east and west coast in particular seem to have a higher proportion of people with that A allele. So that's just one inference I can make. You can make lots more, and this might be one of the traits you choose to look at in the next activity that we do. But this is kind of ticking that first part of the syllabus in explaining how we can look at trends that are based on genetics to learn things about populations. So now we're going to talk a little bit about SNPs, or single nucleotide polymorphisms. And before we go reading through everything that's written on that screen, let's just break down that term, single nucleotide polymorphism. Well, we know single means one, and we should know that a nucleotide is the backbone of the DNA plus a single base. That's what a nucleotide is. Polymorphism we might not be familiar with, but poly we might know means many, and morphism tends to mean form or change or mutation. And so we're talking about single changes in DNA, single nucleotide polymorphisms. And if we have a look at this first dot point down here, it says single nucleotide polymorphisms, which we often called SNPs, are the most common type of genetic variation among people. Pause there. That's important. Most people are genetically very similar. We have very little variation between people, and usually when we have different alleles, eye color, skin color, hair, those sorts of things, usually those are caused by single nucleotide polymorphisms, or very small changes, just a single base in a whole gene, or maybe a couple of bases at different points in a gene. Each single nucleotide polymorphism represents a difference in a single DNA building block called a nucleotide. So we've talked about that already. And here's an example. It says the SNP might replace a C cytosine with the nucleotide T or thymine in a stretch of DNA. We've just changed a base. Could be C to a G, could be A to a T, could be T to a G. We're just changing a single base. So here's a visual representation of a single nucleotide polymorphism. What we should notice if we follow the top of the DNA strand in each of the images, AACT, AAC, AAT, TAAC. There's only one difference. And same with the bottom. So remember, a single nucleotide polymorphism always involves changing only one base on a nucleotide. Now if you look at that picture and you think, well, hang on a minute, there's two changes here, because in the top picture it's C and G, and in the bottom picture it's T and A. Remember that there are two nucleotides present in each of those images. So in that top one, there's the nucleotide that holds the C, and there's the nucleotide that holds the G. And then in this second picture, there's a nucleotide with a T and a nucleotide with an A. So remember that each of those is a different nucleotide, and therefore we can see two different single nucleotide polymorphisms in each of those images. Here's a second image showing another way that you could think about it. This is showing three possible different single nucleotide polymorphisms that a person could have. So potentially this could cause multiple codominant traits. It could be the difference between a dominant or a recessive trait, but most often of all, it'll mean almost nothing or nothing at all, because it's probably not encoding DNA, and therefore it doesn't actually help to build anything in your body. But have a moment to look at that picture. Make sure you can unpack it. Note that all the DNA strands are the same except for that one base, which could be swapped out in any of those ways. Of course, there is a fourth possible option, which is that we put in the C, but I think we've just left that out to make it a little bit smaller of a picture. Okay, so SNPs are not uncommon. You have about 1 every 300 nucleotides, which if you remember how long your DNA chain is, it means that you end up with about 10 million SNPs in the human genome. which means we have about 10 million different points of comparison that we can use to compare the DNA between individuals or to compare the DNA between populations. Most of the time, and I'll just move my mouse around where it says this, most of the time these variations are found in the DNA between genes. That's what we call non-coding DNA, or DNA that doesn't code for building anything. So you might have a gene for your eye color and a gene for your hair color, and between those two sections of DNA on the chromosome, you might have 1, 2, and then a long space of DNA that actually doesn't code for building anything. It doesn't mean that it's totally useless DNA. It serves other purposes, and we'll talk about them a little bit later in the course. But in terms of what effect it's going to have on your body, if it's not used in protein synthesis, it's not really going to affect your body. So most single nucleotide polymorphisms won't affect your body. They are useful for scientists though, as it says down the bottom here. They help us sort out the human genome. Because we know most people share most of their genetics, we can sort of use those as bookmarks to skip between parts of DNA and look for differences in areas that we are focusing on, rather than having to sift through millions and millions and millions of bases. So they're really useful for geneticists in particular, especially if they want to study unusual conditions, unusual traits, or unique gene expression where genes start doing weird things. They can use the known SNPs to find the approximate area where they're expecting to find weird mutations and stuff, and then they can go and investigate a little bit closer and look exactly at what's going on with the DNA bases and things. So we've got a little table here that shows the effects of SNPs. If you've been taking notes along the way, you should have been, I don't think you need to write these down. It's just a good idea to have a quick look at what we might expect when we have SNPs in the gene pool. But that top one is the most common. So this is not the most common one. In a coding region, so this is in a region where we have a gene, there is more than one outcome. Just because you have an SNP doesn't mean that your gene will act differently, because genes use thousands and thousands and thousands of DNA bases and nucleotides, and not all of them are critical. So we can have silent SNPs within a gene. We have an SNP, but it really results in no change in a phenotype. Or it can result in a change in the phenotype. And like it says over on the right here, it might be subtle or it might not. It can be catastrophic. It can cause cancer growth. It can cause terminal illness. More often than not, it causes little or no change. If we're just going to skip, I'm just going to skip these two because they're quite uncommon. It can create a stop codon. That would obviously cause a massive change. It can affect the way transcription happens, and that also would have a massive change, but these are far less common. Down the bottom though, this is the most common. Where we have SNPs or SNPs in non-coding regions, we have no effect on gene products. So it might work as a genetic marker, it might tell us a little bit more about a person's ancestry, but it doesn't actually affect their genetic expression. So the top two are the more likely outcomes if it's in a coding region, and then this bottom one, the most likely of all. So how do we use SNPs? And you're going to need to talk about these sorts of things in a moment when you go off and do your research. One of the first key points is we use it for genetic screening to look for the presence of diseases. So on the screen here we've got a picture of a very common disease in Africa called sickle cell anemia. And sickle cell anemia changes the shape of your blood cells. You can see a normal blood cell here has that kind of round donut shape, and the sickle cell on the right has a very different crescent moon shape, and that significantly impacts on the blood's ability to carry oxygen, because it depletes the hemoglobin and changes the shape, which makes it less effective. And then on the right hand side here you can actually see a picture that shows the SNP responsible for it. So this is the actual SNP. It's just one switch in bases there from an A and T to a T and A, so they've switched positions, which results in the production of the wrong amino acid. And because that is part of a chain that builds up the structure of the blood cell, it changes the shape of the outside of the blood cell and results in that sickle cell shape. A second use for SNPs is in forensics and DNA fingerprinting to solve crimes. So we've got a picture here of what would typically be submitted in a in a court case for a crime where there's DNA evidence. We sample lots and lots and lots of DNA, but there's no point showing most of it because most people share their DNA. So these spikes here are SNPs that have been pre-identified as areas of interest. So if a person has a lot of SNPs that match evidence found at a crime scene, as in this case here, you can see every single SNP is a match, that would tell us with very little error that that person had to be at that crime scene because they have so many of the SNPs. Remember, we have millions and millions and millions of them, and we might sequence 100 or 1,000 in a crime case, and if a person shares all of them, there's almost no chance, like we're talking 1 in a trillion or 1 in a billion at the minimum, of another person sharing it. So we have very strong evidence. So there's no point. The key thing here is that there's no point showing all of the DNA you find at a crime scene from a sample, because you will have, potentially 100,000 bases, and maybe 90,000+ of them are shared by everyone. More, 95,000 more. Who knows? So you focus on the SNPs, and when I say who knows, it's because it depends on what section of the gene, of the DNA. So you focus on the SNP instead. Next one here is looking at human populations, and this is definitely relevant to what you're going to be doing next. So we've got a pretty complex picture on the screen here. We don't really need to break down exactly what it means. What you need to appreciate is that this is an amalgamation of SNPs from populations all around the world, and different colors represent different frequencies of SNPs. And what we can very clearly see is distinct populations in the world. So Africa has a very different genetic makeup than, say, Oceania, where we are, or America, which is very different again. And so we can look at SNPs in populations, and they can tell us about how populations have moved over time, where populations might have come from, especially if we're studying human evolution. We know that humans first evolved in Africa, but the way that they moved across the world is a little bit more mysterious. And you can see, if you look closely, like there's pockets that don't quite match up. For example, some of these East Asian countries like Mongolia and its neighboring countries have a lot of SNPs in common with America. And so that would be something that you would investigate, because it would suggest that there's an overlap between those two populations. And you can see the same thing in Europe. Now, we would expect Europe to share a few SNPs with America because Europeans colonized America. But, this isn't a comprehensive picture of how you go about making these comparisons. It's just showing you that we can see clear differences when we look at SNPs around the world, and those clear differences are important in studying the way that humans have moved throughout history. So I'm going to leave you with this little research task, and I might write a bit more up on the Edmodo post to go with it. Essentially, in order to meet the syllabus dot point, what you need to now go out and do is find some examples of trends, patterns, or relationships in the characteristics of populations. So in terms of this task, what I'm going to get you guys to do is, in your book below these notes, you can just put a little subheading that says something like trends or research into trends. And then what we're going to get you to do is go out and look for some trends either in SNPs or in general population characteristics like blood type, and just write about some general trends. So for example, if you want to take a shortcut and get the ball rolling, you can go back in this video to the section on blood type distribution, or you can Google those graphs yourself, those charts, and you could make a comment about how there are very clear differences in blood types in the population and where different blood types occur. The key point with all of this is that we need to be bringing it back to genetics. So if you're talking about blood types, it's very important that you make it clear that blood types are controlled by genes. Everything that we look at in this next little section should be about genes. So I'm asking you to find 3 examples, at least one SNP. You may have to go into a scientific paper, but I think you'll probably find some if you start looking. You may have even mentioned some already that you can comment on. And if you're not sure about that, about this whole thing and what you're actually doing, go back and have a look at the syllabus dot point, because it makes it very clear exactly what you need to do with the information that I've just given you. Okay, so good luck with that. We'll see how this video goes. Like I said, I haven't really done this sort of a presentation before, so you guys are kind of the guinea pigs. We might do some more in the future when I'm away, or even if you just like this as summaries or something like that. So give me any feedback you've got as well. Other than that, Thank you very much.